So yeah, thanks for listening to this. I'll be talking about population scale transcriptomics, specifically how we are performing RNA-seq on blood from dried blood spots that have been frozen for up to 40 years. Now, as you have probably heard multiple times in this symposium, we have many samples stored in a biobank. Potentially all over the world, there are millions of them. And those samples are very often deeply phenotyped. And together with register-based data, a lot of research opportunities can open up. And this has already been the case for genetic studies. This is like a research resource which has already been tapped for genetics. And the problem for also using it in transcriptomics is that RNA is a very fragile molecule. There are RNAs everywhere. And the general thinking is that you can't really sequence RNA unless you are really, really careful with how you're handling it. And if it spends a couple of hours at room temperature, it will degrade and you can't do anything with it. And we like RNA because it offers some opportunities which DNA will not. It's closer to the actual causal physiology than DNA very often. So in our experiment, what we have done is we have sequenced 554 monozygotic twins, 56 dizygotic twins, using paired end stranded reads and ribosomal deletion. On average, the median age of the sample, so that's how long they have been frozen, is 28 years. For those of you who know something about RNA sequencing, the mean RIN is 2.4, which is actually incredibly low. A lot of protocols will say that you shouldn't even bother with an RNA-seq experiment unless your RIN is above 8. But I think I make a compelling case that that's wrong and you shouldn't actually worry about this. We map 84% of our reads to the genome, which is normal and good for an RNA-seq experiment. The mean quality is good, and compared to samples, the whole blood samples from GTEx, we actually detect more individual genes than GTEx does at every threshold. Now, I mean, there could be problems with sequencing RNA, which is so degraded. And one of them would be, okay, is there some kind of, does it degrade with age? Because if it does, that can introduce biases in any kind of downstream analysis. And that's especially if the degradation is not random, and especially if the phenotype you're investigating is also correlated with age. And here I compare our oldest samples to samples from subsequent years. And you can see that indeed similarity decreases the further away our samples are from the oldest samples in 1981, which is some evidence that, yeah, we see some non-random degradation of RNA molecules. When I performed differential expression analysis using ages of covariate, I found that 33% of all the transcripts show significant evidence for degradation with age. Second problem you can face is that you can imagine that as RNA degrades, maybe some RNA molecules will no longer be detectable. But when I check that, it turns out that no, that's not the case. As a matter of fact, the older our sample is, the more individual genes we can detect, and the more degraded our sample is, the more individual genes we can detect. I have a hypothesis for this, and I think it has to do with us using ribosomal depletion. The reads themselves are only 50 base pairs long. So in a sense, if this RNA molecule degrades into smaller pieces, it's not really a problem. For more details, look at my paper, which will be published soon. Yeah, here you can just see the number of transcripts we can detect in our samples, plotted against the print of the sample and against the age of the sample. And yeah, the older the sample, the better, the more individual transcripts we can detect, and the lower the RIN, the more individual features we can detect. And then to actually see whether this is kind of useful data, we compared similarity of monozygotic twins, dizygotic twins, and unrelated samples. And because transcripts are they're heritable because they are regulated by genes, we would expect twins to be more similar to each other than non-related individuals. And that is indeed what we see. As another sanity check, we also performed differential expression analysis on sex. And we compared our results to results that GTEx published, doing the same thing. GTEx published this list of 500 genes, which are differentially expressed between males and females. And we found that we can detect 229 of them as significant. And also, out of those, 91% showed the same direction of expression, which I think is really impressive, especially when you consider the fact that the samples that we're using come from neonates. They come from babies which are only a few days old, whereas the GTEx samples are from adults. And you can imagine just biologically, a lot of genes will become differentially expressed during puberty. A lot of sex-related genes specifically will only become differentially expressed during puberty. The fact that we can detect so many of them and with such a high concordance, I think is very strong evidence that we have very same data. And so, like I mentioned, we do have this problem of transcripts degrading with in a non-random way. But it turns out we can correct for that using principal components, which is a relatively straightforward thing to do. When I correct the principal components in our differential expression analysis, the number of differentially expressed genes goes down to only 4.3%. And when I try to compare all the samples to all the samples, we see that this effect of it going down just basically disappears. All, like, regardless of how old the sample is, they're about as similar to the oldest samples as everything else. And for those of you who are statisticians, might say, "Oh, you're using principal components correction. Are you just overcorrecting? Are you killing all the signal? Maybe that's why you can't see any differences." And the answer is no. If we look at twin similarity, the actual differences between monozygotic twins and dizygotic twins and unrelated samples will increase. We actually have an increase in our signal after this correction. So not only we're not killing the signal, we're actually magnifying the signal. And the same is true when we look at differentially expressed sex genes. After we do the correction, we can actually detect more of them. And in order to overcorrect, I have to correct for something like 20 principal components. And you can see that all the way at the bottom. When you can't overcorrect and you can't kill the biological signal, but that's not what we're doing. So, yes, so we did manage to sequence RNA from dried blood spots that have been frozen. And we can tell we've been successful because we show very similar results to other studies, and because we can detect differences in the transcriptomes of twin samples. And whereas degradation is real and it is potentially a problem, we find that we can correct for it quite well. 10 seconds before my time. Thank you very much.